proceed. Hi, welcome back,
All right.
everybody, to another episode of METI with a Fantastic Researcher. Today, we have the privilege and honor to have Kevin Yang joining us from Microsoft Research in England. And we are going to have a slightly different format of this talk today compared to the other talks. So Kevin will give us a short intro about generative biology, and then we will follow that by a discussion. Kevin, thank you so much for joining us. We really appreciate it a lot, and very excited to learn from you. The floor is yours.
All right, I'm really excited to be here. Thank you so much. So yeah, today I'm going to be talking a little bit about generative biology, what it is and why we should do it. So a little bit of background is I'm sure most of you know, machine learning has really took over in prediction and in games, right? So first, deep learning really got popular in images, and you can see here over about half a decade, starting in 2011, they really started getting really good at taking an image and figuring out what the image was showing. And eventually, they get arguably better than humans even at some image classification tasks. And that was really driven by this ImageNet dataset out of Stanford. And they're really good at games now. Like if you play Go or you play chess, you know that these neural network-based engines are better than any human. And if you try to beat them on a hard level, you will never beat them because they are superhumanly good. And now the new exciting thing that people talk about is machine learning is taking over generation. We have models like GPT-4 that generates text, Stable Diffusion, which generates images. You can even generate videos now prompted by text. You can describe the video and I'll produce a little generated video for you. And these are all very cool, but there are also concerns about this. Things like who owns AI art? Clarkesworld is a science fiction magazine. And when ChatGPT first came out, they had to close their submissions because people were prompting ChatGPT to come up with these kind of bad science fiction stories, but just submitting hundreds and hundreds of them and overwhelming their system. And when it comes to just generating lots and lots of text, eventually you start polluting the internet with AI translated trash or with AI generated text, which makes the internet a much harder place to find good things. And then, of course, there are also concerns that maybe workers will lose their jobs to AI. And then there's also things like you can generate deepfake porn nowadays much too easily, and people have concerns about that as well. So there's generative AI here. It's very exciting, but also very, there are some concerns. And my perspective on this is that we should really be teaching computers to do things people are bad at. So that includes things like, I've given you the amino acid sequences of some proteins. Can you tell me, and there's some slight differences between them, by looking at these sequences, can you tell me what reactions they catalyze? Can you tell me what differences there are between them? And the answer, unless you've studied these particular proteins, is almost certainly no. This is something that, where the information is in the sequence, but so our human brains just aren't designed to do this kind of learning. And on the other hand, our brains are very well designed to do things like generate text or do art or make music, all right? And we want society to advance, and so society advances when we learn to do things we couldn't do before. We can already make art. We can already generate text. So some examples here are like the steam engine on the left really catalyzed the Industrial Revolution. On the right, we have an example of PCR, the polymerase chain reaction, which is one of the foundational techniques in molecular biology. And so what I want to argue here is that instead of doing generative AI on things that people are already good at, in fact, on things that you can argue were evolutionarily optimized to be good at, in some senses, we should be doing generative AI on things that we're bad at, that humans are very bad at doing, but that would be very useful to humanity and to society. And one area where we do this is biomolecules. And these are especially suitable for generative machine learning for a few reasons. So here I'm showing relative sizes of biology, all the way from atoms to organisms. And sorry, I thought there was something else here, but if we go into really, really small scale, the atoms and such, we can actually do a lot of exact computations. They're expensive. We have to do quantum mechanics. But you can kind of get away with your exact computations. If you go up to the cell, the eggs, the organisms, the questions you want to ask there are very hard to formulate in the precise manners that are good for AI or machine learning training objectives. But if we really focus on that 1 nanometer to 100 nanometer or 1 micrometer length scale, the molecular length scale, here we have often abundant data. We have a well-defined machine learning tasks where we can train very performant models. And my specific focus is at the protein level. And my research goal is to generate new proteins to expand functional space. And what this looks like is we go into nature and we look for natural proteins. And in proteins, there's an amino acid sequence that encodes usually a 3D structure, and that structure mediates some function in the cell. So we can go get tens of millions or even tens of billions of sequences, hundreds of thousands of structures that people have experimentally elucidated. And sometimes we can also know what they do in the cell. And we can train a model that on some of this information to generate new proteins that will hopefully perform new functions that are useful. And that's my pre-prepared introduction. So now we can do a discussion.
Amazing. Thank you so much. This was great. I think it really set the scene for an interesting discussion we could have. In fact, we actually have one more guy here, who is Rasul, I think, also joined us. He's also interested in these topics as well. Right. So, Kevin, let me start by asking you what everybody is fearing today, right? In machine learning and in biology. Do you think we are close enough to an autonomous agent that can start fabricating dangerous viruses, for example? There's a big fear on that happening in society. What's your take on that?
Well, the bad news is there are already autonomous agents that can make dangerous viruses, right? They exist in nature, and that's where we get them in the first place. The other piece of bad news is that if somebody really wants to make bioweapons or to make engineer viruses, people can do that without machine learning, and people have done it successfully without machine learning. Machine learning is not the limiting step. It's not really that hard to make things that are bad in biology. It's much harder to make useful things. But if your goal is to make toxins, your goal is to make viruses. I mean, there's lots of viruses in nature, and we know how to evolve them to make them more dangerous to humans.
Right. So you're saying that it's easier for us to make these viruses in nature than it would be for a machine learning model to make those?
I'm saying though that you don't need machine learning to do this. And that the machine learning does not necessarily give you a step change in people's ability to do this.
Right, I see. Got it. But I mean, okay, so the question that follows out from this, right? So imagine we propose like a drug of some sort using a machine learning algorithm, and then suddenly something wrong happens, right? Something bad happens. It doesn't go well, right? Whose fault is it? Who are we going to blame? What's your take on all those legal or ethical concerns that people are also kind of worried about today?
Right. So when it comes to drug discovery, we already have a framework for making sure drugs are safe and effective, right? And I think the key here, as it often is with many of these ethical issues in machine learning, is, I mean, there's some exceptions, but oftentimes the key is to not be so taken by the new technology that you throw out the existing safety frameworks, the existing ethical frameworks we have, right? So for example, in drug discovery, if your machine learning algorithm proposes a drug that turns out to be unsafe, we have clinical trials, we have safety trials, those we have toxicology trials. Those should catch it the same way that would catch any other drug that we've designed that turns out to be unsafe.
Yeah.
And if for some reason you think your machine learning algorithm is so good you don't need those trials, then whoever decided that should probably be held criminally liable, because that's a terrible decision. But
Right.
the same safeguards in drug discovery that work before machine learning should also work after machine learning.
Okay, interesting. But I mean, are you sort of envisioning a semi-autonomous pipeline in a way, right? Like you're is it fair to say that you have in mind more of a semi-autonomous pipeline where kind of the machine learning algorithm is aiding somewhere, and then at the end it's a human decision to, do things with the output of the machine learning model?
Right. So let's say we have a semi-autonomous pipeline that can optimize molecules for desired property, right? At least for now. I mean, there's some people who would like to go a step further here, but the first step here is going to be a human needs to decide what that property is. All right. What are we going to optimize for? Even assuming you have a very effective autonomous pipeline for doing that. So that's kind of where the human is still in the loop, I guess is a technical term, and really exerts that control and is using this pipeline as a tool to speed up discovery, right? But what gets discovered is still in the hands of the human. It comes from the mind of the human. There are some groups and some people who believe very strongly that we can train autonomous agents on the scientific literature or on other things to also decide what things are worth discovering, right? This is both a much higher technical challenge, right? But also at some level, it only takes a human in the loop up a step further, because at some point you still have to evaluate which of your autonomous agents chose the best things to discover, right?
Right.
And so at some point, it's still a human initiating that algorithm. And I assume if you're initiating the algorithm and you care about science, you're a scientist, that's why you're doing this, you would still check in and see like what has the algorithm decided to go after, right?
Yeah.
Otherwise, part of this goes back to what I'm saying about let's have the humans do what humans are good at and have the machines do what the machines are good at. Humans, so maybe not naturally, but we have taught ourselves and decided it's worth teaching ourselves to be good at discovery, to be good at being curious, and to be happy and to find joy and satisfying our curiosity, right? And in my view, at least, it's very useful to train models and to develop methods to make that process more efficient. But at some point, when scientists do this, they're motivated by that joy of discovery, and I think it'll be very sad, but also very unlikely that scientists will want to take themselves out of that, right? The kind of people who are going to build these models aren't going to want to take themselves away from the joy of discovery.
Right. Fair. Yeah, makes sense. I see. Okay. Right. So I mean, now that we got the, let's say, what worries most of the people out of the way, I think we can discuss a bit more interesting technical questions, maybe, right? So right. So we know that the design process of a drug is very tedious, right? And it's a lengthy process. And there are areas of optimization, right, that we could improve the efficiency of drug design. Could you help us understand what are the major bottlenecks where you think machine learning could actually aid in accelerating a drug design process?
Yeah, so I'm going to caveat all of this with I have never actually done drug discovery before, so most of this is going to be secondhand knowledge. Um, but one of the big challenges in drug discovery is that humans are very complex systems, right? And or that's why we do things like clinical trials as the gold standard there, because these are not standards where we can reason. These are not systems where we can reason from first principle. And there are many, many, many things that can go wrong and that are often hard to think of between thinking this might be a good drug and having a good drug that gets approved and is useful, right? So where machine learning can be useful is there are many steps within that process where you want to predict how a molecule behave under certain conditions, right? And you want to be able to, I guess the term is virtually screen many things, many molecules, cheaply. So, and that's kind of been the first line of use for machine learning drug discovery is, can we do effective virtual screens, basically, to kind of narrow down the design space of things that we want to actually test in the lab. And can we then put that information back in a loop and improve our virtual screens?
Right.
But
Sorry.
yeah.
Yeah, could you say a word for our audience who's not very knowledgeable? How does virtual screening works?
Yeah, so a screen in this kind of molecular biology or drug discovery idea is you have a set of candidate molecules and you're going to measure some one or more properties of them to select the ones that have the property you want, sufficient quantity of a property you want or properties you want, right? And for example, you can take 10,000 molecules and see which ones kill cancer cells on the plate, right? You can take a protein, mutate it a bunch of times, and see which ones catalyze the reaction the fastest or glow the brightest, right? A virtual screen means now, instead of going in the lab and taking your 10,000 molecules and seeing which ones kill the cancer cells, you've gotten some data on that, you've trained a model, or you have what you believe is a first principles model that's not trained in data. And you're going to take a bunch of hypothetical molecules and score them using this model. And then maybe you take the top 1% or the top 10% and try them in the lab. And the hope is that makes you it makes your screening much more efficient. The flip side of that, though, is that you can also make an argument that the effort you put into that is better put into making the screens themselves faster and cheaper. So you can just get more real data. All right. At some point there's a trade-off.
I see. Okay. Right. So if I understood you correctly, it's like kind of, we have a lot of molecules, right? And then we build a model, and that model predicts the properties of these molecules. And then from that, I can filter out what molecules I would like to, for example, manufacture if I trust my model, something like that.
Yeah, usually it's way upstream to manufacture. It's you virtually screen billions or millions of molecules. You take the top 1 or 10%, and you try those in your cells, and you try those in the lab. And then you verify your virtual screening hits with a real screen.
I see.
And then you take those candidates and move them to the next thing.
All right, got it. And I mean, therefore, building the model is actually critical for the success of virtual screening, right? And what are the major caveats of, just standard machine learning application to kind of predicting properties of molecules? Can I just plug in a graph neural network out of the box and it works, or are there any issues that I need to solve first?
All right, so machine learning typically has two ingredients, right? You have your model and your training algorithm, but you also have the data you train it on. And both of those are important, right? For example, when it comes to predicting molecular properties, there has been a lot of progress in what model architectures have inductive biases that are suitable for learning on molecules, because molecules have these properties like they're atoms with bonds between them, there's rotational and translational invariances that you can make the training much more efficient by building some of that information into the model, for example. And there's been the field of geometric deep learning is really concerned with that. On the other hand, there's also a lot of effort into things like, if I want to, for example, test predict the ability of family of small molecules to bind to a protein, right, which is an important task in science and drug discovery. And I train a model that does, I get some data set from some public data set where people have measured whether this molecule will bind a specific protein. And you train a model on it, and you fill out a validation set as one does, and a test set, and you do very well. And you say, great, I have an amazing model. Let's go use this downstream. The problem is then you go look at the data you found online and you find a few problems. One is that it's collected in different labs and they all measure things slightly differently. And each lab cares about their own protein, right? So now you have this confounder where every protein, the binders of every protein are measured differently. You also have this problem that you have different binders measured against different proteins. When you have many protein, often you have many binders against one protein, but there are many proteins where you have very few binders, and then you find that your model does very poorly when you try to extrapolate the proteins that aren't in your training set. So the data is very important, and a lot of times we're using legacy data that wasn't meant for statistical learning. And there's a lot of things that to be careful about depending on where you want to apply your model.
And I mean, is data collection like a tedious? I mean, I suppose data collection could be a tedious process, right? And in machine learning, we have these ideas of semi-supervised learning and contrastive learning, where we do like sorts of data augmentation, right, to kind of grow my data set, so I don't need to collect much more data than what I already have. Are there any ideas that carry over to biology or to molecules in general? Could I somehow do some sorts of data augmentation to increase the size of my data sets or so?
Right. So data augmentation makes sense when the augmentation teaches the model about symmetries in the data, right?
Right.
And that's really where the information gain is coming from. Just making random perturbations to your data isn't actually going to help, right? Unless those perturbations teach it about something important. So in biology, people try to, who are most familiar is going to be with proteins. Like I have a protein, I want to predict what it does, either in a classification sense, like this is X class of enzyme or Y class of enzyme, or this variant of this protein will give you more turnovers on this reaction, etc. The difficulty here is if we knew the invariances in sequence space, we would have solved the problem, right? We wouldn't be training models. We could just do it from first principle. And the problem often with trying to do data augmentation here is that we often don't know the invariances that you can use to augment the data. Now, self-supervised learning actually has been fairly successful in biology and in proteins specifically. So in language, most people did things like, they started with things like word to vec, and then eventually they found they did BERT with transformers, where you corrupt your sentences and then reconstruct them, and that lets you do some pretty good. It improves performance in a lot of downstream classification prediction tasks, right? People, of course, then applied this to proteins because there's sequences of amino acids, so you can treat the amino acids like language tokens and do word to vec or doc to vec or BERT. And so word to vec or doc to vec works okay, but then where it really took off was with these BERT style models, where people would take you would go look for millions or tens of millions of publicly available sequences, and you can do this corrupt and reconstruct task with transformer models even. And this worked very well for a few reasons. One is that arguably transformers are good at text, but they're actually really, really well designed for this in proteins. And that's because if you want to predict this three-dimensional structure of a protein, right, from the sequence, what you really want to extract in your self-supervised stage is which positions in the protein interact with which other positions, which is exactly what attention does, right? And there are lots of near close interactions, obviously, in sequence space, but it's also really important to pick up the few long-range interactions. And transformers are really well equipped to do that because they consider every position and with every other position explicitly. And so transformers are like the perfect architecture for doing this in some sense. And because you can, without any supervision, you can go look at the attention maps or perturb the inputs and get out pretty accurate, remarkably accurate correlations between positions that are close together in a three-dimensional space. And for example, the new state-of-the-art structure prediction models like AlphaFold do actually have a sequence reconstruction self-supervised module, and that looks in your sequence database basically, and it teaches the model about what positions that the biology termers co-evolve, right? They change when one changes, the other one tends to change with it, and that tells you that they're probably close together in three-dimensional space. Transformer cell models are really good at picking that up, and then the big advance with AlphaFold is really, instead of what previous models had done, is they try to infer this contact map, and then they train a model to refine the contact map, and then they had a not learned folding module that predicted the structure from this refined contact map. The innovation in AlphaFold 2 was really learning that folding module end to end. So you started from your sequences or alignments, you did the self-supervised stage, and then you backprop signal from the 3D structure all the way back to the beginning. And that was really the innovation there through the folding module.
Right. Okay. Is it in some sort trying to learn a kernel between different protein structures? Like, is that the main question in a way? Because I mean everything that goes back to invariances, right? And anything in machine learning, really, from my point of view, is just trying to learn a sort of similarity structure which I can use to predict things in the future, right? And the question is it true that's what all is happening actually? Isn't that everything? If we have a good kernel, right, between two proteins, I can do anything I want, or is there more to it than that?
Well, having a good kernel between protein sequences is a non-trivial problem. But I don't want to try to reason off the cuff about kernels for like structured output spaces, because that seems tricky. But if we go back to what I really focus more on, which is function prediction, I have some sequence. I would want to predict the outcome of some measurement of that sequence based on measurements I've seen before. So there's a lot of early work here from me and from from a guy named Philip Romero of the Arnold Lab on, we want to do Bayesian optimization here. So we use Gaussian processes, as one does, on the sequence space to try to get good predictions with uncertainties to do GPU-CB and efficiently sample the space to find sequences with optimal properties, right? And the algorithms we kind of were all self-taught, or at least I was self-taught, so I just kind of apply these things naively with standard kernels. We tried a few kernels like your squared exponential or a matern kernel. And they actually, in the lab, you make the sequences, you find really cool things. You find molecules that do things you couldn't do before. But then we actually looked at the predictions later, and we thought about these things later. And we observed first empirically that the uncertainties are actually very miscalibrated, which tells you that the kernel you're using does not reflect the actual function space, right? That maps from sequence to measurement.
Yeah.
Right. That not only is the true function not in your prior, but there's probably nothing even remotely similar to the true function in your prior. And mostly it's just the case that doing any learning at all in these kind of machine learning guided direct evolution experiments will always be not doing any learning, but it's actually very hard to improve on the first thing you try. Even if the first thing you try isn't actually that great. And then later under some theoretical work out of Deborah Mark's lab, where they showed that these standard kernels actually don't work very well for biological sequences, and mathematically why, and how they are not flexible enough in the ways they need to be flexible for discrete sequences.
Right. I see. Interesting. Yeah, this is interesting. Would we also see that if we were operating in a latent space of a transformer, for example. So what I mean is, say we take the input sequence, right? And then we pass that through a transformer, so we encode it, and then we go to the latent embedding of the transformer, and then we actually define a GP on top of the latent embedding. Will we still also suffer from the same issues?
Yeah, so I've definitely seen papers that do that, where they, like I said, you do your Gaussian process space optimization in the latent space. And they also, of course, they're published, so they report good results. I have not dug into them myself to see how well calibrated they actually are. It'd be an interesting experiment to do to see if putting them in latent space actually does give you nice functions between input and output that you can model with GPs with standard kernels. Yeah, it's hard to say without digging into that, digging either deeper into the math or empirically testing some of these claims.
Yeah, I mean, this is very interesting that you mentioned this because, like we had a paper, right, about this, we called it antigen BO, right? So, but I think we observed something very similar to you, that whatever we did in simulation, so to say, and whatever is in the real world, there's kind of a big gap between them. The question, I mean, this is an interesting question because in robotics, right, we actually have this problem. We call it the sim to real problem, right? Where we work in simulated worlds and the robot is different, so we need to bridge that gap, right? But I haven't seen people in, like, say, biology or molecular design actually attempting, to see how I can bridge this, let's call it sim to real gap of biology or something, right? Where, I work in my simulator and I want to learn something and then take that into the real world somehow, right? How do you envision, like this to be closed, this gap to be closed? Because we need simulation, right, to accelerate designs, right?
Yeah, the trick is your simulations need to be based on real data, right? And so if we're talking about optimization, the challenge really is that you want to optimize over non-trivial sequence landscapes, right? So if the sequence space you're considering only has 100 points, just make them all, and then you have the global optimum now, right? So you really want to be optimizing over spaces where you can't do that. The problem is, if you're optimizing over spaces where you cannot physically measure things, and we haven't solved the problem of predicting in these spaces, right? That means it's really hard to get ground truth landscapes to play with. All right, so the first one of these that's sufficiently big to be interesting came out when I was in grad school. And what they did was they collected most of the variants at 4 locations in this protein. So that's 4 locations, 120 amino acids, 160,000 locations, right? There's some caveats to the quality of the data, but it was large enough to be really useful for testing these sort of optimization algorithms. Because that's a big enough landscape that for most properties, you wouldn't want to actually enumerate and test everything. So now what the consequence of that is, I like to say that every protein model-based optimization or Bayesian optimization paper is overfit to that data set. Because that's the one we have, right? And now there are definitely some efforts out there to systematically make public some more of these data sets on systems where it's tractable, but also interesting, and where you might hope it applies to systems where it's more difficult. All right. But yes, at some level, if you don't know the first principles, even at a very expensive, even with infinite computation, and you don't have ground truth optimization landscapes, it's really, really hard to compare methods. All right. The other thing I guess you could do is take our methods and go actually make them in the lab and compare them head to head. That doesn't require you to elucidate the complete landscapes. That is also fairly high effort. And then there will be a lot of politics about whose algorithm gets to be tested, right? Why mine got left out.
Interesting. Yeah, to follow up on that, I wanted to ask you a question because I've been toying with this idea for a while. I don't know what you think about it, but in again, this is inspired by robotics research, right? But in robotics research, we do a lot of domain randomization, right? When we want to bridge this sim to real gap. So
Hmm.
kind of the idea is that we start with our system, and we don't learn a controller or a policy just on that system. But what we typically do is we kind of randomize, the dynamic parameter of the system, and then
Hmm.
we try to find a policy that works well on average, right, over all those randomizations, with the
Right.
hope that this transfers better to the real world, right? This is kind of the
Hmm.
idea. Um, could I do something similar in these fields of biology? Like imagine I have simulator, which typically has some parameters, right, which are set up, either by us or from real data or something. How about if I run an optimizer, but not only over one instance of that simulator, but over a distribution, right, of
Right. Right.
simulators? Would that be more robust to like simulation mistakes I would make?
Right. So there is also a lot of work on can we. It's really hard to go get complete optimization landscapes, right, for protein sequences. So
Mm,
can we come up with synthetic ones that are realistic enough to be useful? And then yeah, synthetic ones are going to have knobs you can turn. You can sample the knobs, the knob positions, and then you get a distribution of landscapes, and then you can test things from those, right? Uh, the problem there is again, no, I'm not saying you couldn't do this, but no one that I know of has actually aligned those to the
mm,
real world to show that if you do well in their suite, you will do well in the real world. And that's what people really would want to see to use those in practice. All right. Um,
right.
and there's people proposed various simple to complex versions of these. Uh, There's an old one, the original one's called like the NK landscape, where you control how much epistasis there is between, how much interactions there are between positions. The thing, popular, easy thing to do now is like you have some real data, you train a neural net on it, and you call that your oracle. Uh,
Mm,
but yeah, no one's really tested these simulated landscapes or oracles to see if performance on that correlates with performance on real systems either.
I see. Got you. Okay. All right. Interesting. Yeah, also we have Rasul here. I don't, Rasul, if you have any questions, also please go ahead. I've been bothering Kevin a lot already.
Hi, hello guys, can you hear me?
Yeah.
Yeah.
Okay, cool. First of all, thank you, Kevin, for this brilliant talk. It's very interesting. Yeah, my question is, one of the questions that I have is that, like, if we're talking about a drug discovery, right? And as I understand the process of drug discovery, it's a lengthy, quite expensive, and complicated process where, like, even when you come up with some protein sequence, yeah, the testing of that in order to get like the answer on the question how useful it can be kind of like quite long and quite expensive. And that's why people
Mm-hmm.
try with some animals. They firstly tested that on some animals. Then basically after, if like the test on animals kind of like was confirmed that it's okay, they start to use it on these voluntary human participants actually. So my question is
Mm-hmm.
this: like, as I understand that, when we try to evaluate the performance, like, or the behavior of this protein on human, the amount of data you will collect on human participants typically also will be quite small. Like, it's not like we will be
Yes.
basically, like, we will have an access to millions of people who will basically voluntarily decided to participate in that. So
Mm-hmm. Mm-hmm.
how this apparently a small size data regime when it comes to machine learning models kind of like affect the way the whole process of finding the sequences, because eventually this number or the value that we'll get, how good it is for human beings, it will be very noisy based on the fact that you only use very small portion of like people, like, or yeah. So how it's actually, I guess it's basically the question of like how to how to basically improve the fact, how to overcome the fact that you eventually evaluate on a very, very small number of people in order to get the value which will be exactly the value that you're looking for, like the most important one. Like,
right. So I think there's a few questions here. One is in general, the current state of the art is, well, I guess you could call it traditional statistics for like power analysis, right? What kind of sample, how big of a sample do I need to have so much, what degree of confidence that my molecule is safe, basically, or that my molecule is effective at treating the thing I want to treat, right? So that's a very well-studied field. There is still ongoing research there in things like, in what cases should I stop a trial early because it's obviously effective or obviously ineffective or dangerous, right? In what cases do I need to deal with, is it important to deal with subgroups in my population and in my trial, and how do I deal with that? So those are active areas of research still. The other side of this is, are there things we can do to predict the effect of a molecule in a population of humans or a specific human, right? Based on something we can do in a lab or in a computer, which is also an active area of research. And it's a very challenging problem. For example, one line of research here is, for example, is there a way to map from experiments I do in cell culture and the effect in the cells there to what the effect will be in the whole organism, in mice or in humans, right? And can I do that on a population scale? Or if I have cells from a specific person and I take them out of their body and put them in the lab and do it, obviously that's not the same environment. So there's not going to be a one-to-one exact match between the effects, but can I learn from data how to predict from the effects outside the body to the effects inside the body? And that's also an open question, whether that's possible. If it is, it would obviously be very useful. So those are the things. Once again, drug discovery is not my specialty. There are people who do nothing about drug discovery, but these are the things I've heard about in the field.
I see. Thank you. And the second question I have, I don't know, maybe it's kind of like, maybe it's in some way funny question, is that if we, for example, if we know that we have to deal with in the circumstances that the number of people of like participants, human participants will be quite small, can we somehow benefit from that very big corpus of data we got when we tested on the animals? Like how basically that generalizable from the animals to the humans? So could we somehow efficiently reuse that data? Like, it's like there's no any strong correlation.
To some extent, we already do that, right? If we're testing a drug, first you make sure it's safe in mice, and then you make sure it's safe, maybe some other model organism, then you make sure it's safe in humans, right? With the expectation there is that if it kills some mice, you probably shouldn't put it in the people. Going beyond that, it's challenging because there are differences between mice and people, and also the mice can't tell you how they feel, right? And the mice care about a lot less things than people do, at least we think. So yeah, that's really, and part of this is why I was saying when it comes to generative biology, I think the low-hanging fruit is really at the molecular level before you get to the organisms. Because here, if my goal is to generate molecules that do a well-defined thing in a vial or in a reactor, that's a much simpler system than a molecule that cures a disease without causing other problems. Because the reactor is a system I can control in pretty close to 100%, whereas the human body is not a system I can control close to 100% with any sort of efficacy or ethics.
Thank you very much. Yeah, I have no questions. Thanks. It was a great talk.
Kevin, I still have 2 more to go, and then I'll let you go.
All right.
Okay.
Wonderful.
So the first one is more of an advice, right? So, working in the field that you work in is actually very difficult because it involves, a couple of expertise, right? So it involves chemistry, biology probably, and chemistry. And then it also involves computer science and machine learning and AI, right? So it's really tough, right? Because, people with this expertise are hard. So the question here is, what's your advice for chemistry and biology people who would like to enter into the machine learning world? And what's your advice for the machine learning people who would like to do, molecular type of work? How should they approach these problems? Maybe you can tell us a bit more about your past, your history, how you got that. Maybe we can all learn something from it.
Yeah, so I like to joke that I convince the CS people that I'm a biologist and I know things about biology, and I convince the biologists that I know things about CS, so that no one really sees how little I know. But the more serious answer here is that in general, a deep technical understanding in one field prepares you for deep technical understanding in other fields, right? Does not mean you automatically have that understanding, but you should have the confidence and the skill that from learning one field, and those meta learning skills are applicable to the next field. For example, when I'm interviewing candidates, if I see a candidate that has limited machine learning background, but is interested, but they have really deep, really solid knowledge and experience in another technical field, whether that's biophysics or chemistry or anything else, I can be fairly confident that they can learn what they need to do a deep learning project, right? So if you already have some deep domain knowledge in chemistry or biology or anything else, and you are interested in the neural networks and machine learning side, there's kind of two ways that people can choose to learn this kind of area, right? You can do breadth first search or depth first search.
Right.
Right. So breadth first search says, I want to go down all the avenues and make sure I kind of progress everywhere at once. And for people who really want to understand the whole field and who don't like having holes in their knowledge and who really don't want to do anything they don't understand, that can often be a very good way to go. And obviously, the startup to finishing your first project is going to be a little slower. So depth first says, from my existing expertise, I have this idea I'd like to work on. I'm going to learn the things I need to do that project. All right. I have an idea. This is my goal. This is the outcome, the model I need. I've done a little bit of research. I've talked to some people. I know this is a kind of machine learning. These are the machine learning things I need to do that. I'm going to learn those really well. I'm going to dive deep into that project. And then the next project, I'll pick something. I might do something different. All right. And my personal taste is to do a little bit of the breadth first, just so you know, like the basics, and won't spend too much time going down unproductive rabbit holes and trying to do impossible things, like physically impossible things. But then I just find that motivating things with a specific project is much more fun, right? If you have a specific task you're trying to do instead of just building toy models for the sake of learning. There.
Got it. Okay. Interesting. Okay. All right. So kind of like explore a bit, and then gain the basic building blocks, so to say, and then try to then decide to go deep a rabbit hole at a certain moment.
Yeah.
Okay. Fair. Interesting. Okay. Right.
And then I think part of it there too is to pick problems that your existing expertise really lets you work on. All right. So if you're a biologist, I would not recommend coming in and trying to do state-of-the-art like image generation from images you just create from the internet. Just because one, that's a very popular field that's very well funded and people are working on it. And why do that to yourself? right? Two, because I don't think that's generally super societally useful or interesting thing to do for
Right.
reasons I've outlined earlier. But also because you have valuable training in the field already, right? You know what the problems are in that field better than almost anybody else, I hope, right? Uh, why not solve the problems that only you can, that you're uniquely positioned to solve?
Yep. Yeah. I see. Yeah, that's that's very valuable. I think you're right. It does make sense. It's interesting because I also seen many of my friends trying to make a jump, which is very similar to what you just said. And yeah, it's a good advice. Like you're really good at this thing, yeah, and nobody else knows it. So why don't you just do it? Like you're the
Yeah,
expert, right? Yeah. Okay.
and to an extent, especially in academia, being
Yeah.
the first one to define a problem that turns out to be important is really nice because your
Yeah.
attempt to solve the problem is probably not going to be the best attempt ever, but you still get credit for being the one to define the problem,
Yeah. You created the
right?
problem. Exactly.
Yeah.
Yeah.
And you don't
Fair.
have the pressure of having to like beat 10, 100 previous attempts.
Yeah. There's no baseline.
Yeah.
Right. Makes
You
sense.
get to be the baseline.
Makes sense. Interesting. Right. And now the last question. This is more maybe more of kind of another advice, so to say. But in machine learning, right? We publish a lot of papers. There's a lot of papers coming out. Was it in drug design, biology, new architectures, whatever you name it, right? And today there's big hype about large language models, as we know, right? And vision language models and diffusion models, right? So how do you keep track of the literature? Like I for once finding it really hard, to actually keep track of what's happening every day because there's like so many papers. What's your is there a strategy you follow for that?
Yeah. So mostly like everybody else, I drown in the literature. The strategy I've settled on, for better or for worse, is you want to have like an awareness of what's going on in general, right? You cannot possibly read every paper, and you definitely can't deeply understand every paper, right? So whether it's through social media or through like an RSS feed or something, you want some small amount of time just being aware of what's out there, right? And then you want to pick some papers that you find interesting or particularly relevant to read all the way through. You want to find some papers maybe a little bit less relevant or less interesting where you just kind of skim it or look at the abstract. And then there's going to be a few papers where you really need to understand the top to bottom. And part of the hope here is that as you get very familiar with the subfield, you can skim a paper and quickly situate it in like, this is what they're doing. This is how they build on what's done before. This is what the thing they do that's new. And this is how these are the things they're claiming. And here are my subjective opinions about how well grounded those claims are in the evidence they presented. And for papers in my subfields, I can do that very quickly and very efficiently. If you ask me to do it for something outside, then I can do it, but it takes a much more effort, right? Because I don't have the background down quite as well. But that depth in a particular field takes time, and there's not really a substitute for thinking deeply about a field and reading deeply about a field. Right. But at the same time, you cannot think and read deeply about every field. Right. And I think it's good to have an awareness of the broader, what's going on more broadly, just for ideas like I have a problem. What are some possible things? Can I take inspiration from this thing that seems maybe unrelated? And that's where the awareness comes in. The other side of this is hopefully you have collaborators and colleagues who have some similar interests, but also some different interests. And when you talk to them, hopefully they know some things that you don't know, and they can point you to useful things.
Yeah. Okay. Sounds good. All right. Interesting. Yeah. That's another good advice. Awesome. So man, yeah. I really appreciate you being with us. I think it's really interesting. I know you're always on the look for good people to join your team in Microsoft, right? So this is also an opportunity if anybody's interested in what Kevin has been doing, which I think is awesome, right? So do feel free, I guess, to reach out. Is that fair, Kevin?
Yeah, so typically most of the reach out that I get are people interested in students interested in internships. So and the thing I feel the worst about is when someone who I would love to interview and possibly make an offer to emails me right as we finish intern hiring for the year. Uh,
That
so,
hurts.
but yeah, so our cycle is we usually open applications in like late October for internships starting the next summer. And we try very hard to have offers accepted by sometime in January. Sometimes we get additional allocation later than that, but that's like the bulk of the allocation.
Right.
There's also an undergrad program which is centralized through MSR North America.
Uh-huh.
This year there's 30 undergrad research interns in all of North America, and somehow the BioML group at Health Features and Microsoft Research New England has, I think, 3 of the 30 because because we are enthusiastic about hiring interns here.
Sounds good. All right, no problem. So yeah, Kevin, I'd like to thank you again, really, for taking the time to talk to us. And really, I mean, I follow you a lot. I follow all your work. I think it's always fantastic and a lot of fun to read.
Thank you.
So yeah, and with that, I would like to end this talk with Kevin and ask our audience to like, share, and subscribe to the channel. And I will see you another time for the next time in Meti with a fantastic researcher. So thanks again, Kevin, for coming. We appreciate it.
Thank you so much.
Cheers.